Now we are going to read chapter number 15 of NCERT Biology Part 1, that is Plant Growth and Development. You have already studied the organization of flowering plant in chapter 5. Have you ever thought about where and how structures like roots, stems, leaves, flowers, fruits and seeds arises, and that too in an orderly sequence? You are by now aware of term seed, seedling, plantlet, mature plant. You have also seen that tree continuously increase in height or girth over period of time. However, the leaves, flower and fruit of same tree not only have limited dimension but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in plant? All plant organs are made up of a variety of tissue. Is there any relationship between structure of cell, a tissue, an organ and function they perform? Can the structure and function of these be altered? All cells of plant are descent of the zygote. The question is then why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of mature plant from zygote, fertilized egg, follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that proceeds roots, leaves, branches, flowers, fruits, and seeds, and eventually they die. The first step in process of plant growth is seed germination. The seed germination, when favorable condition for growth exist in environment, the absence of such a favorable condition, the seed do not germinate and goes into period of suspended growth of rest. Once favorable condition return, the seed resume and metabolic activities and growth takes place. In this chapter, you shall also study some of factors which govern and control this development process. These factors are both intrinsic (internal) and extrinsic (external) to the plant. 15.1 Growth. Growth regarded as one of the most fundamental and conspicuous character of living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part or even of an individual cell. Generally, growth is accomplished by metabolic process both anabolic and catabolic that occurs at expenses of energy. Therefore, for example, expansion of leaf is growth. How would you describe the swelling of piece of wood when placed in water? 15.1.1 Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity of unlimited growth throughout their life. His ability of plant is due to presence of meristematic at certain location in their body. The cell of such meristem have capacity to divide and self-repage. The product, however, soon loses the capacity of divide. Such a cell make up the plant body. The form of growth wherein new cells are always being added to the plant body by the activity of meristem is called the open form of growth. What will happen if the meristem ceases to divide? Does it ever happen? In chapter 6, you have studied about the root apical meristem and shoot apical meristem. You know that they are responsible for primary growth of plant and principally contribute to the elongation of plant along their axis. You also know that dicotyledon plant and gymnosperm, the lateral meristem, muscular cambium and core cambium appear later in life. These are meristems that cause the increase in girth of organ in which they are active. This is known as secondary growth of plant. 15.1.2 Growth is measurable. Growth at a cellular level is principally a consequence of increase in amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to its. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cell in watermelon may increase in size up to 350,000 times. In the former growth, the express as increase in cell number. Later, express growth is as increase in size of cell, while growth of pollen tube is measured in terms of its length, and increase in surface area denotes the growth in dorsiventral leaf. 15.1.3 Phases of growth. The period of growth is generally divided into three phases, namely metameric, elongation, and maturation. Let us understand this by looking at tip root. The constantly divided cell both at root apex and shoot apex represent meristematic phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nucleus. Their cell walls are primary in nature, thin, cellulosic, with abundant plasmodesmata connections. The cell proximal to the meristematic zone represent the phase of elongation. Increase in vacuolation, cell enlargement, and new cell wall deposition are the characteristics of cell in the phase. Further away from the apex, that is, more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. The most of the tissue and cell type you have studied in chapter 6 represent this phase. 15.1.4 Growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of organism that produce more cell in variety of ways. Here below there is diagrammatic representation of arithmetic growth and geometric growth and stages during embryo development showing geometric and arithmetic phases. On next page, the growth rate shows an increase that may be arithmetic or geometric. In arithmetic growth, following mitosis cell division, only one daughter cell continues to divide while the other differentiate and mature. The simplest expression of arithmetic growth is exemplified by root elongation at a constant rate. Look at figure 15.5 on plotting the length of the organ against time. The linear curve is obtained mathematically. It is expressed as LT is equal to L naught plus RT. LT represents the length at time t. L naught represents length at time 0. R is equal to growth rate hyphen elongation per unit time. Let us now see what happens in geometric growth. In the Morse system, the initial growth is slow in bracket lag phase and it increases rapidly thereafter at exponential rate in bracket log or exponential phase. Here both progeny cell follows mitotic division, retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slowdown lead to stationary phase. If we plot parameter of growth against time, we get typical sigmoid or S curve. The sigmoid curve is characterized of living organism growing in a natural environment. It is typical for all cell, tissue and organ of plant. Can you think of more similar examples? What kind of curve can you expect in a tree showing seasonal activity? The exponential growth can be expressed as WL is equal to W naught E power RT. WL is equal to final size, W naught is equal to initial size, R is growth rate, T is time of growth, and E is based on natural logarithm. Here R is the relative growth rate and also the measure of ability of plant to produce new plant material referred to as efficiency index. Hence, the final size of WL depends on initial size of W naught. Quantitative comparison between the growth of living system can also be made in two ways. First, measurement and comparison of total growth per unit time is called absolute growth rate. Second, the growth of given system per unit time expressed on a common basis. Example, per unit initial parameter is called relative growth rate. In figure 15.7, two leaves A and B drawn that are different sizes but show absolute increase in area in given time to give leaves A dash and B dash. However, one of them shows much higher relative growth rate 1 and Y. 15.1.5 Conditions of growth. Why do you not try to write down what do you think are necessary conditions for growth? This list may be have water, oxygen, nutrients as well as essential element of for growth. The plant grow cell grow in size by enlargement which in return requires water. Turgidity of cell helps in extension growth. Thus, plant growth and further development is intimately linked to water status of plant. Water also provides the medium of enzymatic activity needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients in bracket macro and micro essential element are required by plant for synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be determined to its survival. Environmental stages such as light and gravity also affect certain phases stage of growth. 15.2 Dedifferentiation and redifferentiation. The cell derived from root apical and shoot apical meristem and cambium differentiate and mature to perform specific function. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell wall and protoplasm. For example, to form a triarchy element, the cell will lose their protoplasm. They also develop a very strong, elastic, lunicellular secondary cell wall to carry water to long distance. Even under extreme tension, try to collate the various anatomical features you encounter in plant to the functions they perform. Plants show another interesting phenomena, the living differentiation cell that by now lose the capacity to divide and regain the capacity of divide under certain condition. This phenomena is called as dedifferentiation. For example, formation of meristem interfascicular cambium and core cambium from fully differentiated parenchyma cell. While doing so, such meristem tissue are able to divide, produce cell once again, lose the capacity to divide but mature to perform specific function, that is get redifferentiation. List some of these tissue in the woody dicotyledon plant that are product of dedifferentiation. How would you describe a tumor? What would you call parenchyma cell that are made up of to divide under controlled laboratory condition during plant tissue culture? Recall in section 15.1.1 we have mentioned that the growth of plant is open, that is it can be indeterminate or determinate. Now we may say that even differentiation in plant is open because cell tissue arising out of the same meristematic having different structure in maturity. The final structure at maturity of a cell tissue is also determined by location of the cell within. For example, cell position away from root apical meristem differentiated as root cap cell, while those pushed to periphery mature as epidermis. Can you add a few more examples of open differentiation collateral? The position of cell to position is an organ. 15.3 Development. Development is a term that includes all changes that an organism goes through during its life cycle, from germination of seed to sensation. Dramatic representation of sequence of process which constitute the development of cell of higher plant is given in figure 15.8. It is also applicable to tissue organ. Plants follow different pathway in response to environment of phases of life to form different kind of structure. This ability is called plasticity. Example: heterophyll in a cotton, coriander, and larkspur. In such plant, the leaves of juvenile plants such as different in shape from those in mature plant. On the other hand, difference in shape of leaves produced in air and those produced in water in a buttercup also represent the heterophilous development due to environment. This phenomenon of heterophilous is an example of plasticity. Thus, growth, differentiation, and development are very closely related events in the life of plant. Broadly, development is considered as the sum of growth and differentiation. Development in plant, example, both growth and differentiation is under control of intrinsic and extrinsic factor. The former includes both extracellular, genetic or intracellular factors, chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrients, etc. 15.4 and apparently most important part of this chapter is plant growth regulators. 15.4.1 Characteristics. The plant growth regulators are small, simple molecule of diverse chemical composition. They could be indole compound in bracket indole 3 acetic acid, IAA, or adenine derivatives, that is nitrogen 6 phosphorylate amine purine, kinetin derivatives of catecarbonides, abscisic acid ABA, terpenes in bracket gibberellic acid GA3, or gases in bracket ethylene. The plant growth regulator are variously described as plant growth substance. Plant hormones are phytohormones in literature. The PGR can be broadly divided into two groups based on their function in the living plant body. One group of PGR are involved in growth promoting activities such as cell division, cell enlargement, patterns formation, trophic growth, flowering, fruiting, and seed formation. These are called plant growth promoter. Example: auxin, gibberellin, and cytokinin. The PGR of other group play an important role in plant responses to wound and stress of biotic and abiotic origin. They are also involved in various growth inhibition activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. This group PGR ethylene could fit in either of group but largely an inhibitor of growth activities. 15.4.2 The discovery of plant growth regulators. Interestingly, the discovery of each of five major groups of PGR have been accidental. All this started with observation of Charles Darwin and his son Francis Darwin when they observed that the couples of canary grass respond to an unilateral illumination of growing toward the light source in bracket phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of entire coleoptile. Auxin was isolated by F.W. Wendt from tip of coleoptile of an oat seedlings. The Baccanin in bracket foolish seedling. The disease of rice seedling was caused by fungal pathogen Gibberellin fulcorni. E. Kurosawa 1926 reported the appearance of symptoms of disease in rice seedling when they were treated with sterile filter of fungus. The active substance were later identified as gibberellic acid. F. Skum and his co-worker observed that from internode segment of tobacco stem the callus in bracket mass of undifferentiated cell perforated only if in addition to auxin The nutrients medium was supplemented with one of following extracted vascular tissue, A. Extract coconut milk or DNA. Skoog and Miller later identified and crystallized the cytokinin promoting activity substance that they term chitin. During mid 1960s, their independent research reported purification on chemical character of three different kind of inhibitor: inhibitor B, abscessin 2, and dormin. Later, all three were proved to be chemically identical. It was named as abscisic acid (ABA). Cousins confirmed the release of volatile substance from ripened organ that hazarded the ripening of stored unripened banana. Later, this volatile substance was identified as ethylene, a gas plant growth regulator. Let us study some of physiological effect of these five categorized PGR in next section. 15.4.3 Physiological effect of plant growth regulator. 15.4.3.1 That auxin. Auxin, from Greek auxin, to grow, was first isolated from human urine, and term auxin is applied to indole 3 acetic acid and other natural and synthetic compounds having certain growth regulation properties. They are generally produced by growth apical of stem and root, from where they migrate to the region of their action. Auxin, like IAA and indole butyric acid, have been isolated from plant NAA and 2,3-D. 2,4-D are synthetic auxin. All these auxins have been used extensively in agriculture and horticulture practices. They help to initiate rooting in stem cutting and applicable widely used for plant propagation. Auxin promote flowering. Example: in pineapple, they help to prevent flower and leaf drop at early stages but promote the abscission of older mature leaf and fruit. In most higher plants, the growth apical bud inhibit growth of lateral axillary bud, phenomena called apical dominance. Removal of shoot tip usually result in growth of lateral buds. It is widely applied in tea plantation, hedge making. Can you explain why? Auxin also induce parthenogarphy. Example: in tomato, they are widely used as herbicide. 2,4-D, widely used to kill dicotyledon weed, does not affect mature monocotyledon plant. It is used to prepare weed-free lawn by grass gardener. Auxin also control xylem differentiation and help in cell division. 15.4.3.2 Gibberellin. Gibberellin are another kind of promotory PGR. There are more than 100 gibberellin reported from widely different organisms such as fungi, higher plant. They are denoted as GA1, GA2, GA3, and so on. However, gibberellic acid GA3 was one of the first gibberellin to be discovered and remains the most intensively studied form. All GAs are acidic. They are produced which range of physiological response in plant. Their ability to cause an increase in length of axis is used to increase the length of grapes stalks. Gibberellin cause fruit like apple to elongate and improve its shape. They are also delay sensation. Thus, fruit can be left on tree longer so to extend the market period. GA3 is used to speed up malting process in brewing industry, beverage industry. Sugarcane store carbohydrate as a sugar in their stem. Spraying sugarcane crop with gibberellin increase the length of stem, thus increase the yield by a much as 20 ton per acre. Spraying julienne conifers with GAS has turned the maturity period, thus landing to early seed production. Gibberellin also promotion bolting in beet, cabbage, and many plant with rosette habit. 15.4.313 Cytokinin. Cytokinin has specific effect on cytokinesis and were discovered as kinetin from autoclaved herring from sperm DNA. Kinetin does not occur naturally in plant. Search of natural substance with kinetin like activity led to isolation of zeatin from corn cells and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinin and some synthetic compound where cell division promoting activity have been identified. Natural cytokinin are synthesized in region where rapid cell division occur. For example, root apical, developing shoot bud, young fruit, etc. It helps to produce new leaves. Chloroplast in leaves, lateral shoot growth and adventitious shoot formation. Cytokinin help in overcome apical dominance. They promote nutrition metabolism which help in delay of lip sensation. 15.4.3.3 Ethine. Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amount by tissue undergoing sensation and ripening fruits. Influence the ethylene on plant include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedling. Ethylene promotes sensation and absorption of plant organ, especially of leaves and flowers. Ethylene is highly effective in fruit ripening, enhance respiration rate during ripening of fruit. This rise in rate of respiration is called as respirational climatic. Ethylene breaks seed and bud dormancy, initiates germination in peanut seed. Sprouting of potato tuber. Ethylene promotes rapid internode petiole elongation in deep water rice plant. It helps leaves upper part of shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping plant to increase their absorption surface. Ethylene is used to initiate flowering for so-synchronization fruit cell. In pineapple, it also includes flowering in mango. Since ethylene regulates agriculture, most widely used compound as source of ethylene is ethiopian. Ethiopian is an aquatic solution, is readily absorbed and transported within the plant and release ethylene slowly. Ethiopian has fruit ripening in tomato and apple and accelerated abscissions in flower and fruit. It promotes female flower in cucumber, thereby increasing the yield. 15.4.3.5 Abscisic acid. Abscisic, as I mentioned earlier, abscisic acid ABA was discovered for the role in regulation, abscission, and dormancy. But like other PGR, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and inhibit of metabolism. ABA inhibit seed germination. ABA stimulate closure of stomata in the epidermis and increase tolerance plant to various kind of stresses. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps seed withstand decision and other factors unfavorable for growth. In most situations, ABA acts as antagonist to GA. We may summarize that for any and every phase of growth, differentiation, and development of plant, one or other PGR has some role to play. Such role could be complementary and or antagonist. This could be individualistic, or synergistic. Similarly, there are a number of events in the life of plant where more than one PGR interact to affect event. Example: dormancy in seed, bud, abscission and sensation, apical dominance, etc. Remember, the role of PGR is only one kind of intrinsic control along the genomic control and extrinsic factor. They are an important role in plant growth and development. Many of extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of light and temperature on initiation of flowering. 15.5 Photoperiodism. It has been observed that some plants require periodic exposure to light to induce flowering. It is also seen that such a plant are able to measure the during of exposure to light. For example, some plant required express to light for periodic exceeding a well-defined critical duration, while others must be exposed to light for predict less than this critical duration before the flowering initiates in them. The former group of plants are called long day plant, while the latter one term short day plant. The critical duration is different for different plant. There are many plants, however, where there is no such a correlation between exposure to light during an induction of flowering response. Such plants are called day neutral plant. It is also now known that not only duration of light period but that duration of dark period is also equally important. Hence, it can be said that flowering in such a plant depend not only on combination of light and dark exposure but also really relative duration. This response of plant to period of day night is termed as photoperiodism. It is also interesting to note that while shoot apical modify themselves into flowering apical period to flower photoperiodism, the site of perception of light dark duration are the leaves. It has been hypothesized that there is a hormonal substance that is responsible for flowering. This hormonal substance migrates from leaves to shoot apical for induction flowering only when plants are exposed to necessary induction photoperiodism. 15.6 Vernalization. There are plants for which flowering is either quantitative or qualitative, depend on exposure of low temperature. This phenomenon is termed as vernalization. It prevents precocious reproductive development late in growing season and enable the plant to have sufficient time to reach maturity. Vernalization refers especially to promote of flowering by period of low temperature. Some important food plant, wheat, barley, rye have two kind of variation, winter and spring variety. The spring variety are normal plant in the spring come to flower and produce grain before end of growing seed. Winter variety, however, if planted in a spring, would normally fail to flower and produce mature grain within a span of flowering season. Hence, they are planted in autumn. They germinate and over winter come out as small seedling. Resume growth in spring and have harvested usually around midsummer. Another example of vernalization is seen in biennial plant. Biennial are monocarpic plant that normally flower and die in second season. Sugar beet, cabbage, carrot are some of the examples of biennial plants. Subjecting the growth of biennial plant to cold treatment stimulate subsequent photoperiodic flowering response. Seed dormancy. There are certain seeds which fail to germinate when external conditions are favorable. Such a seed had understood to be going a period of dormancy, which is controlled not by external environment but are endogenous control or controls within the seed itself. Impermeable and hard seed coat. Presence of chemical inhibitors such as abscisic acid, phenolic acid, para-ascorbic acid, and immature embryo are some of the reasons which cause seed dormancy. This dormancy, however, can be overcome through natural means and various other man-made measures. For example, seed coat barrier in some seed can be broken by mechanical aspiration using knives, sandpaper, etc., or vigorous checking in nature. These aspirations are caused by microbial action and passage through digestive tract of animal. Effective of inhibitory substance can be removed by subjecting the seed to chilling condition or by application of certain chemicals like gibberellic acid and nitrate. Changing the environment conditions such as light and temperature are other method of overcome seed dormancy.